Hello and welcome to today's webinar. On behalf of Drug Target Review and Halo Labs, I'd like to thank you all for attending. I'm your moderator, Nikki Withers, and I'm the editor of Drug Target Review. Today's speaker will be Gabriel Mercado, who's the Senior Marketing Applications Scientist for Halo Labs. Following his presentation, we will move on to a live question and answer session where you can pose questions to today's speaker. Please remember that you can submit questions at any point during the webinar using the questions panel, which is situated in the menu on the right. So without hesitation, may I pass you over to today's keynote speaker, Gabriel Mercado.
Hello, everyone. Very nice to be here. It's a very nice foggy morning here in San Francisco, and we have a great talk for you, so I hope you enjoy these results. We're going to talk about small molecule solubility screening and a solution that Halo Labs has to make your life easier in the lab. Okay, in the content, I'm going to be framing the problem. The problem is the solubility of small molecules and how this affects drug discovery efforts. And then I'm going to tell you about the solution that we are establishing here in the company and how this has been a great aid to many scientists and collaborators already. I'm going to walk you through some of the results we got here at the company. And then for the conclusions, I'm going to pinpoint the advantages of using this instrument for this endeavor. So a little bit about me. I am a biophysics by training, and I have been engaged in several drug discovery efforts. And drug discovery is not trivial, it's a big endeavor, and then solubility is always a present problem at many stages through the drug discovery process. Let me show you. This is a graph of the scientific publications trying to capture the interest about solubility or aggregation in the title. And to me, it's not a coincidence that at the same time that the combinatorial chemistry libraries became available to researchers, and they started doing these high throughput drug discovery protocols. That's at the same time that there was a renewed interest in solubility and aggregation, because a lot of these new chemicals, a lot of these new chemical entities had solubility problems that produce a lot of headaches. In particular, I'm going to be talking about false positives and also the limited bioavailability of these compounds. The false positives affect the early drug discovery processes, in the discovery and the preclinical development, and these are a common artifact. Aggregates produce artifacts that take over a lot of the resources and time from the researchers. I have two examples here. This affects everything from cell systems to biochemical coupled systems to pure biophysical assays. Aggregates can form these colloid molecules, or they can form the macromolecular colloid aggregates, and these will actually activate molecules in the cell system, or will activate off-pathway systems, and they will produce either false positives or false negatives. Even when you look at the SPR, for example, aggregates produce very, very particular signals that are indistinguishable from a real hit. In this case, for example, it was a compound that either look irreversible or look reversible, and they can even show dose response behaviors. They are very, very credible hits until you, for example, in this case, put a little bit of detergent that helps solubilize the chemical, and then the effect goes away. Right now, if you're doing a drug discovery and you're at this stage, you have to start studying about the solubility of your compounds. Otherwise, it will lead you into this wild goose chase, and at the end, you will have spent a lot of resources in something that is just a beautiful artifact. Formulation is the other point where solubility is very important, and this is throughout the clinical development stage of drug discovery. Solubility is very important, but seldomly attained in new drugs. Right now, about 40% of the drugs in the market belong to this class 2 of low soluble drugs that, although they are low soluble, they have a high permeability, which is a good thing. It's calculated that 60 to 70% of the current drug prospects in the market will also belong to the same class 2 low solubility class of drugs. And this is where formulation is going to take place, and is going to be a very important part of the strategy to bring these drugs into the market. Most drugs can be solubilized if you have a good system to quantify solubility. And one thing of interest is that in this class, you have a phenomena that if you see crystals, this becomes a dissolution rate limited process that is actually, unless you want something that is rate limited dissolving inside the body, you don't want this. You don't want crystals. Crystals are a kind of an energetic solubility trap, and they don't allow for compounds to solubilize very easily. So if you see amorphous colloids, you are solubility limited, but at least in this space here, this is called the formulation space. Using formulation, you can improve the solubility of your compounds to try to make them more drug-like. So this is how the phenomena looks at the molecular level. This is what you want. You want monomers of your new chemicals, free-floating, homogeneous monomers. But in reality, a lot of the drugs are going to start forming oligomers. They are going to initially be dimers, trimers, but they can even start piling up and form huge oligomers. That eventually will produce these insoluble aggregates or even the crystals. This is what you want to look into. You want to see, is this happening to my molecule? So it is very nice in the visible, there's no problem, because if you have one of these chemical drug libraries, you just have to look at the plate and you can immediately see which ones are precipitated, which ones have crystallized. So just by eye, you can already see a lot of these compounds. But not everything that looks transparent, not everything that looks clear is free of aggregates. And that's why people have invested a lot of money and resources trying to find ways to better be able to observe and characterize these aggregates. So what is being done mostly of the time is that they use molecular technologies, for example, dynamic light scattering or mass spectrometry, NMR, or powder X-ray crystallography, for example, to try to observe these phenomena here. The problem is that these current tools that are out there, they take a lot of time. They are better suited really for the molecular level. So they have a lot of problems. For example, if the media is not homogeneous, like in dynamic light scattering, you are not able to identify and characterize types of aggregates. Everything looks like an aggregate and everything looks like a huge aggregate. You cannot differentiate between types of aggregates. Techniques like, for example, size exclusion chromatography require you to filter the sample. and then you immediately lose all the aggregates that you had there in the subvisible. So these techniques are very nice as an orthogonal way, but before getting here, one thing that is highly overlooked is the subvisible. The subvisible is that part where a solution is clear but might still have aggregates, and it's very easy using just microscopy and backgrounded microscopy to look at these aggregates, be able to characterize them. You can immediately tell from amorphous precipitates from crystalline precipitates, and this is a throughput way to get information before you go to the more sensitive molecular devices, right? So this is the part that is missing right now in the equation. Turbidimetric and nephelometry, they do cater at this level, but they are not sensitive enough, and they require a lot of your precious material, up to 150 microliters of your sample just for an N of 1. So a need was found for this subvisible space, and when we were looking into this, this is the solution that we came up with. We built this Horizon system that uses the background methodology, and it's a very convenient solution. I brought with me a lot of my drug discovery projects to the company, and I was immediately charmed by the small volume that the machine needs per sample to be able to detect these colloidal aggregates. It's amicable to automation. You know how important that is during drug discovery. You can look at the subvisible without a problem, from 2 microns to 4 millimeters. And in this space, there's a lot of information, a lot of things that just by looking at them allow you to make a better experiment. And then this is fast. You can, if you have a small library, you can certainly look at it in the course of 1 or 2 days. And the way this instrument works is very nice. What it does is that you have this filter plate here. We call it the background plate. You have to take a background image of this plate using the device. And then once that is loaded into the instrument, what you do is now you load the sample into these wells. As I said, as low as 25, but you can load more. Then there's a manifold vacuum that aspirates the sample, basically eliminating all the liquid, everything stays dry, and then all the particles remain there. You then take a sample image of that, and then the instrument, what it does is that it subtracts all the background from the background image, and then immediately things become very obvious. Sensitivity increases many-fold, and you get a clear picture that the instrument then, using its onboard analytics, immediately starts processing for you. And then it will give you not only, for example, the image of every single particle it detects, but it will also give you analytics, for example, histograms or scatter plots that will help you understand what's going on with your sample. We're going to look into an example of this later. So I'm going to talk a little bit about what we did. In this case, we use kinetic solubility measurements, but you can also use, for example, equilibrium solubility samples to bring to this point. And the way we did it is that we started serial dilutions of the compounds we tested on pure DMSO. So we did the stock solution, and then we kept diluting that in DMSO. From there, we diluted 100-fold in PBS, so that the DMSO was 1% final. This is very typical of a chemical library. And then we analyze these samples either using the Horizon instrument or spectrophotometer methods, turbidimetric methods, the very same sample. So this is what we saw, and I'm going to let the results speak for themselves. In the first case, we have these four compounds that we've tested. These are available drugs that are in the market right now. And as you can see, as you increase the concentration of the compound, you see different kinetic solubility profiles for these molecules. Over here, we're following percentage of area cover. So 100% doesn't mean that it stopped precipitating. It just means that we reach saturation of our membrane. These compounds then, as you can see, at some point they start coming out of solution, and then they have different ramps. They have different rates of precipitation. This one, for example, TITP precipitates faster than the other ones at much lower concentrations. So this one is the most insoluble of our compounds. In contrast, this compound, diclofenac, was soluble at all concentrations tested, even at 1 millimolar concentration. So this is very useful. This is was done in a matter of minutes, and it already gives you a profile, a kinetic solubility profile for your molecules. That is something that is very important. If we go into detail here, in this part here, where we're still, the points are still sitting on the asymptotic level, what we see is that we set up a threshold of solubility. Anything before, below 0.5% of the area cover is just going to be baseline, right? And I'm not showing here, but if you go backwards, you just see a baseline that basically you are at the noise level. But then, as soon as you reach a concentration where your compound starts coming out of solution, anything that touches this threshold, you will immediately see a jump, like a trigger jump of a material coming out of solution and forming the rest of the curve here. So this is interesting. TIPT, for example, starts precipitating at the same time, at the same concentrations as dipyridamole, but the rate of TITP to become insoluble is much higher than the one for dipyridamole. So this is very interesting. They kinetically, they fit different exponential equations, and this is already information that is very valuable. In the case for glycofulvin, as you can see, it only starts precipitating at higher concentrations around here, and then it starts going up. We didn't go higher, but this either would plateau here or maybe will continue going. It's something we have to research. And this is the case for diclofenac. As you can see, at all concentration tested, it never reached the threshold. So we deem it soluble at all concentrations. But how does this compare with these more traditional subvisible methods of looking into the aggregation of these particles? So what I'm doing right here is I'm comparing now our data that we took with the Horizon instrument with the data that we obtained spectrophotometric by a turbidimetric method. And as you can see, the order that we perceive precipitation, the coming out of solution, is the same between the both methodologies, but you can also see that the horizon was much more sensitive. And this makes sense because you need a lot of material to start precipitating before you can observe it using a spectrophotometer. But when using a microscope, when you can count individual particles, anything that rises above the particle threshold, you will immediately be able to detect, even if it's 2 or 3 particles above your threshold. And that's how you can become more sensitive. And as you can see, then this methodology was able to give us a better range of concentration usefulness for each of these compounds. Although the order was the same. So if we put everything into a table like this, this is the solubility range that we measure for these particles. We're putting, between 10 and 20 micromolar is the solubility range, because this is the range where the compounds seem to be coming out of solution. Just to give you a number then, I just took the mid-range value for this and put it here. And if we do the same for the turbidimetric assay, what we see is that we're like 10 times higher sensitivity than the turbidimetric methods. Although the solubility rank order, as I've said before, stays constant. So this is very important because if you're using turbidimetric, let's say you are using Roche's LISA, for example, which basically they also follow a type of turbidimetric assay. This will give you an estimate kinetic solubility range that is going to be off of the reality. So this is better. It's better to know that things are precipitating earlier, because this range between 15 and 150 could cause artifacts that you would think are real hits, and they are not. And this is the second part that this instrument was very valuable for us. One of the things you can see is the morphology of your precipitates. And morphology of the form, it's very important because it not only tells you the phase of your compounds, but it can also point to ways to solubilize these compounds. For example, in this condition where we treated dipyridamole, we could see crystals, right? These kinetic traps, so these are not good. But there are other conditions where you would see a different kind of precipitate. In this case, these flexible needles, right? They didn't look crystalline, but we also found ways that these ones could be dissolved. Or you can see very amorphous material, that in the case for regression of fulvene here. All this is important because if you remember, what you want to see if you can do the formulation to improve. So in this case, it would be hard to do formulation, but in this case, it would be easier to have a formulation so that you can put your drug into the market. This is a solid form transition that we observe. So when you are doing the formulation, let's say that you find the crystal form and you ask yourself, is there a formulation that can stop the forming of crystals so that I can have a better bioavailability of my drug once it enters the body? And you can certainly design experiments where you can follow this. In this case, for example, for regression of fulvene at 25 degrees incubation, at these concentrations, we originally see some amorphous particles that just by leaving the tube on the bench after about 12 hours, what you would see is that there would be an increase of the amorphous. This is to be expected. This is already talking to you about the bench life of the compound at room temperature. But if you just leave it long enough, a whole day, then something very interesting happens. You see a drop in the number of particles, and then you also start seeing these crystal forms appearing. And this is very useful because this is already telling you that this drug will go through a solid form transition into crystal. And this is something that was that is usually overlooked. So this would be a great system to be able to look into this, and it's amicable to be aromatized because the horizon gives with its onboard onboard analytics, it's already primed to do that. Crystal forms are geometric entities. In this case, for example, this one is longer, is long but thin, and the crystals are bigger than the amorphous particles, so you find a smaller amount of particles in the membrane. This is something that you can determine. So if you look, for example, into geometric parameters, for example, here the ferret, where you measure the length against the width of your particles, you will see that all the crystals would cluster together like this, while the amorphous particles are more spread. If we look at an amorphous samples, for example, this one that you see this small precipitate all over the plate, then the analytics, this same type of graph, parameters graphs here, will just show a spread of all these particles in ferret and in circularity, for example. This data is readily exported from our system, and it can be put through scripts to detect these type of things automatically, so that you would be able to know, for example, crystals from amorphous particles automatically, right? So this is very useful. And then let me just go to the conclusions here. I think, in all my experience in drug discovery, that this is a real solution in the early discovery of drugs and also during the formulation of the drugs. It's flexible and it's really easy to use. We train junior biochemists, and in less than 2 hours they are monitoring samples and they are producing results. Our system is more sensitive than turbidimetry, and it's definitely your first choice that you should use before taking to other orthogonal tests where you want to see at the molecular level what's happening. It's faster, easier, it uses less volume. These are dry measurements, so there's no matrix interference. Many of the methodologies that are out there right now, the matrix, the buffer, the conditions of the solution of your compounds definitely affects what you observe. We don't see that at all. These particle images that you obtain using these instruments provide you with valuable information of the solid state format we saw. And this is crucial for formulation. We think that this should be used as a primary solubility screening method, and it will help you eliminate problematic candidates faster. And that's very important, again, because you could have your heart set on a particular lead, and this lead turns out to be false. So the faster you eliminate bad leads from your lead repertoire, the better off you're going to be. And this certainly can be implemented in any BMI technology or early ADME workflows. And it's certainly a step up in the laboratory analysis and automation technology to be able to discriminate into what exactly is going among all these chemicals you are testing. And with that, I hope you enjoyed those results. I hope I was not very fast. And please, if you have any questions, this would be the time to answer them.
Thank you, Gabriel, for that excellent presentation. So we now have the pleasure to introduce our question and answer session, where today's speaker will answer all of your questions. Don't forget, you can still submit your questions using the questions panel in the menu on the right. So let's take a look at the first question. So, Gabriel, how do results correlate with other techniques?
Well, you just saw the correlation with the turbidimetric method, where we showed that the rank order is the same. So if you are doing rank orders, our instrument will give you the same rank orders as other techniques. But at the most sensitive range within the subvisible. So if other techniques can also, you can take them to correlate to the subvisible, we will be able to give you the same rank order, although the exact concentration at where something starts to become an oligomer, for example, we might not be able to see, but we will be able to tell you when these soluble aggregates come out of solution, for sure.
Perfect. Thank you, Gabriel. And so our next question, what are the material and time expenditures for the BMI technique?
The BMI technique is, once you have your instrument in place, there's only one consumable that you need, and this is the plate. The plate is basically a filter, you saw it, where you apply your samples. You use one well per sample, and then this you can pass to the manifold where you do the aspiration and then take into the instrument, and that's all you need. You can do only one well in your plate and then use the rest of the plate in other experiments. So the only expenditure right now is the cost of the plate. That if you want more information, please send us an email, and I'll put you in contact with the best person to answer more details about this.
Thank you, Gabriel. So our next question, does SM color interfere with the analysis?
It doesn't. That's a big thing. In many instruments, if your chemical compounds have a color, it will be interfering. For example, some chemicals are fluorescent. So if you are using a fluorescent technique to monitor aggregation and they fluoresce, then this gives you data that is very, very complex and hard to interpret. In our case, that's not the case. We have two types of illumination where we resolve the particles, bright field or side illumination. And with these two, it's very easy to, for example, determine the types of aggregates. If something is gel-like or oily, it immediately becomes apparent. Because most of the time, what you could have is that it's not your chemical that is aggregating, but it's causing the aggregation of, for example, an additive. Using this instrument is very easy to know the difference.
Perfect, thank you. And moving on to our next question, does the form of the precipitate give information about the nature of the precipitate?
Yes, it does. As you saw, for example, we saw some crystalline forms, and these are chemical entities that are very difficult to resolubilize. And for example, you want to stay away from them in case that you want to bring this to the market. But if you have an amorphous precipitate like the one we were looking at, then this would be easier to formulate. At least there would be a possibility to be able to formulate so that to be able to increase its bioavailability. And for example, if you have a system that consists in a protein forming fibers, let's say alpha-synuclein or one of these, and you can observe these fibers, then you can follow what's the response of the protein in response to your drug.
Perfect. So we've got a few more questions here for you. Next one is, can the horizon be used with equilibrium solubility samples?
Yes, they can. This is an analytical method. So the way you prepare your samples, either kinetic or equilibrium based, or any other way you want, you just bring them, and then this instrument will be able to analyze them for you. And it certainly would be a good idea to compare kinetic from equilibrium results using the device.
Lovely. And next question here, can this method be used for pre-screening solubility?
Yes, it can. It could be very expensive because some people have libraries that consist of many thousands or sometimes even millions of chemical compounds that they use to do drug discovery screening. But if after you get these results, you have a concerted library of chemicals that are leads. You can also use these at this stage, even before going to other orthogonal techniques, so that you gain more information and be able to eliminate immediately all those leads that had solubility issues.
Perfect. And we've had another question in, Gabriel, any experience in applying the technique for judging solubility of new agrochemicals?
Agrochemicals, no, but I cannot see why it would be different. If you have a model system and you are already testing for these agrochemicals, then it would be just a matter of translating that into the device as an analytical tool for you to get those results.
Perfect. Thank you. And so that is the end of our webinar today. So thank you, Gabriel, and for the audience, please join me in thanking Gabriel Mercado for his time. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. So please take a moment to provide your feedback. If now's not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Halo Labs, I'd like to thank you all for attending today's webinar, and we hope to see you again soon. Goodbye.